All right, we're going to go ahead and check out the tube media for microbiology. Our next tube of interest is going to be TSI, triple sugar iron. This tube has a butt and a slant portion. When we inoculate with the needle, we stab the butt and streak the slant. We are testing not only for sugar fermentation, but also for the presence of hydrogen sulfide, utilizing the iron in TSI. Possible results for TSI is that the entire tube turns yellow. If the entire tube turns yellow, that's an indication that all 3 sugars were fermented. Along with all 3 sugar fermentation, a purely yellow tube, you can also have a positive result for hydrogen sulfide. Just like in the motility media, the hydrogen sulfide appears as a blackish precipitate at the bottom of the tube. Another sugar result for TSI is where you have the butt that's yellow, but the slant remains red. This is an indication that only glucose was fermented. Notice that there are some breaks in the agar. Many times these organisms will ferment with gas production. What that does is push the agar around the tube. That's not an uncommon result, but we still read the colors the same way. Yellow in the butt, red in the slant is an indication of glucose fermentation only. In addition to sugar fermentation, you can also determine whether or not your organism is positive for hydrogen sulfide. In this case, we have a yellow butt, red slant, that tells us about glucose, and we also have a black precipitate, which gives us an indication of hydrogen sulfide production. It is positive. All right, the next tube we're going to be looking at is thioglycolate. Thioglycolate is testing for anaerobic versus aerobic growth. So a tall column, sort of a champagne pink color. We're looking for growth primarily at the surface, primarily at the bottom, or anywhere in between to differentiate obligate from facultative anaerobic. An organism that grows throughout the entire tube, so we see growth, turbidity at the bottom, at the top, in the middle, in any combination, that organism is a facultative anaerobe. It can grow with or without the presence of oxygen. A tube where we see growth primarily at the surface, primarily at the top, that's an indication that the organism has to have the oxygen. That's an example of an obligate aerobe. And then when we see growth at the very bottom of the tube or closer to the bottom of the tube in heavy growth, that's an indication that the organism is a obligate anaerobe. And our last media of interest is OF dextrose. Dextrose is a form of glucose. These are always paired tubes. We have one tube that is exposed to the environment and one tube that we added sterile mineral oil to produce an anaerobic environment. It's a nice green color when uninoculated. Green means the organism is not able to oxidize the glucose, the dextrose, or ferment the dextrose. Negative. An organism that can both oxidize and ferment will turn OF dextrose yellow in both the aerobic and the anaerobic tube. This is positive. And then our final possible combination for OF dextrose is it's able to oxidize in an aerobic environment, but it cannot ferment in an anaerobic environment. Yellow-green, positive-negative for OF dextrose.